pleased today. حق الميكرو ولا سي بون؟ So I'm pleased today to share some essential tips and tricks with you for effective cell culture management from media preparation to cryopreservation. So the presentation will begin with the basic needs of cell, followed by key aspects of cell culture, including seeding, passaging, cryopreservation, and handling contamination challenges. So how to maintain a cell in culture? As you know now, cell culture is the process of growing and maintaining cells outside their natural environment, thanks to the culture media that provide nutrients like amino acids, vitamins, minerals, energy sources to maintain optimal physicochemical conditions such as neutral pH, and to support cell proliferation with specific growth factors. There are two main types of culture media, natural or artificial. For natural media is less controlled and more variable, with including biological compounds directly derived from tissue extracts or bodily fluids. In contrast, artificial media or synthetic media are carefully formulated and adapted for specific cell types. They are often named after their developers, like Eagle, Parker, etc. Media can also be classified based on the complexity of their composition as either basal, like DMEM, or complex, like RPMI. Media formulation must often be supplemented with serum, which is a biological fluids with a variable and uncontrolled composition. It's often used at concentrations ranging from 2 to 20%. And there are various types of serum, including horse serum, calf serum, newborn calf serum, and fetal bovine serum, the most used one, and the most rich and the most expensive too. So serum contains several important compounds. Growth factor boosts cells to divide, while hormones support specific cellular function. Attachment factor helps cells stick to the culture surface, and protease inhibitors prevent damage from enzymes like trypsin. Serum also provides basic nutrients and other compounds that provide cell protection. However, serum can have safety issues, so there are alternative mixture available to replace it. This is an example of culture of primary human dermal microvascular endothelial cells. We compared here two mediums, DMEM F12 and a specific endothelial cell medium. We compared also two supplements here and here. So when with 10% FBS, There was no significant difference in growth between the media, here and here. I don't know if it's clear. Okay. There are no difference with a minimal progression observed from day 1 to day 3. In contrast, when we switch it to a combination of 5% FBS and a specific growth factor supplement, cell proliferation increased markedly here and here. In addition, in the specific medium, we observed that cell growth reaching 100% confluence compared to 70% with DMEM F12. This experience highlights the importance of selecting a suitable medium and supplement mix, especially for primary culture, to achieve optimal results, of course. Let's move now. Let's move on the different stages of cell culture process. For cell seeding, tea flasks are commonly used to provide space for cell attachment and growth, while Petri dishes and microwave plates are chosen based on precise cell requirements. I think everyone know now that cells are maintained in a complete medium at 37 degrees with 5% CO2. But it's important to monitor factors like medium color for pH balance, for example, to replace medium frequently, and to observe cell density and morphology. Here you can see the cells at different stages of growth: 10%, 30%, 50%, and 90% confluence. A microscope allows us to make a visual estimation of cell growth over time. So we don't need only a regular microscope. The best is an inverted microscope. Le contraste de phase. Voilà. When cells reach 70 to 20%, it's time to passage them to maintain a healthy growth. So first, remove the old medium, then wash with PBS and add a trypsin solution to detach cells. You can incubate briefly at 37 degrees Celsius. You can also gently tap the flask to assist with detachment. Once done, neutralize the trypsin with PBS, mix gently, collect the suspension, and centrifuge for 5 to 10 minutes. After that, resuspend the cell pellet in fresh medium, count the cell, and seed an appropriate volume in fresh medium and in a new flask or dishes. That's it. Let's continue with the cryopreservation. Cryopreservation is crucial for ensuring a stable cell supply, limit genetic death, and maintaining standardized cell line through cell bank. For optimal condition, there are 3 important factors. So first, choose a good cryoprotectant. I think the best is DMSO. Second, include a protein source to support cell integrity. Third, perform a gradual freezing at a rate of -1 degree per unit using a special container. Use that to prevent ice formation in the cells. This is the protocol. So put the cell pellets, let's see, about 4 million, I think, at least it's I think 4 or 5 million of cells. So put the cell pellets on the ice and resuspend it with cold, it's very important, cold freezing medium. Then distribute the suspension in the cryotubes and put them in the special container at minus 80 degrees Celsius and leave them overnight. The next day, perform rapid freezing by immersing them in the liquid nitrogen. For cell thawing, the important tips are thaw cells quickly, immediately discard the freezing medium, and replace the medium after 24 hours of seeding. Let's conclude with the constraints, mainly contamination. So in cell culture, we face two main types of contaminants, chemical and biological. For chemical contaminants, they come from materials around the cells, like detergents, free radicals, metal ions, plastic residues, water impurity, etc. Biological contaminants include bacteria, yeast and mold, virus, and mycoplasmas. Here there are some examples of different types of biological contaminants in cell culture. On the left, you can see microbial contamination, often visible under a microscope. On the right, we have mycoplasma contamination, which can't be seen with a regular microscope and needs special tests for detection. So to prevent contamination, we often use antibiotics, and there are several types routinely used. For example, penicillin, streptomycin. Specific kits may also be needed, such as for mycoplasma elimination. Unfortunately, antibiotics alone isn't enough, as most contamination is caused by improper handling. So before concluding, here there are essential tips for proper cell culture practices. Don't share your solutions. It's very important. Prepare a liquid of stock solution. To preserve the stock solution and to prevent contamination, cryopreserve cells routinely. Avoid touching your hair, your face, and your phone while working. Keep your incubator and the water bath clean. Maintain a clean, cool lab environment. Wipe surfaces, hoods, and gloves with 70% ethanol frequently. And limit external items in the lab, dusty boxes, stock material, and dirty gloves. That's it. Thank you for listening.